we were honored to present our work. It represents combining the information from three different biomarkers that can be measured in the cancer tissue, typically from a biopsy. Two of these markers are already well known in the cancer field. One is called MKI67, and it's typically used to track proliferation in the cancer cells. And another marker is very well known in the cancer immuno-oncology world, and it's called PDL1. And it's typically a biomarker that people have used for a while now to try to understand whether at least one major mechanism of immune evasion is already at play in the cancers. And the reason people have used this marker is to try to predict whether treatment with certain immuno-oncology drugs, like inhibitors of PD1 or PDL1, will have efficacy in terms of tumor shrinkage or even complete tumor disappearance, either on imaging or pathology after such treatment has been completed. The problem has been that no technique till date has really been accurate or accurately predictive of such efficacy. And this is a big need, a big requirement, a big unmet need, if you will, in the field, because until such accurate biomarkers are available and accessible, we really will not be able to further improve the rates of success in the clinic when using drugs like this. So our approach was to try to introduce a third biomarker in combination to try to improve the performance of these predictive biomarkers. And the third biomarker that we introduced is a transcription factor called FOXC1 or FOXC1. And it is not primarily tracking proliferation like MKI67. It is primarily tracking a process called plasticity. And plasticity is a property that cancer cells acquire typically as they get more advanced in their life cycle, where they are able to change, morph into different shapes if it helps it spread to other organs. It helps it acquire chemoresistance, radiation resistance, and even resistance to immune therapy drugs. And so, the hypothesis going into this was that if we can track all three major properties of a cancer that are known to affect efficacy, we may do better in our prediction efficacy for such drugs. So that was the basis for the work. And we basically trained our model on one clinical trial data set from the I-SPY2 trial, which was a breast cancer neoadjuvant trial. And then we tested our prediction algorithm accuracy on two other clinical trial data sets, so for validation, in other words. And the results were quite promising and was received quite well by the audience. So we're encouraged to continue our work in these cancers as well as other cancers.